So, so in a diploid organism, so here is your, here are your homologous chromosomes over here, fine. They are genes all across the length of this chromosome, okay. And if we take any chromosome from here, chromosome 11, chromosome 11 has 3000 genes, okay. So if this is chromosome 11, homologous chromosomes, one comes from mother, one comes from father, this chromosome will have 3000 genes. And if we label them from one end to the other, gene 1 will be here, gene 2 will be here, gene 3, gene 4, gene 5, gene 6, all the way till gene 3000. Okay. The same genes are also in the homologous chromosome, all the way to 3000. You will have in this location over here the same gene in both the chromosomes. So basically, we have two copies of the same gene in these homologous chromosomes, which are in the nucleus and which are inside the cell in our bodies. Now, if these two copies can be identical or can be different, either way, they are alleles of the same gene. So for a diploid organism, which has two homologous chromosomes, there will be one gene because the information is the same, but there'll be two copies, and the copies are called alleles. Is that clear?
Okay, so means gene is a fundamental of a fundamental part of a chromosome, and allele is a group of that gene.
No, I wouldn't put it like that. I would say that in a single stretch of DNA, which is here, there is a physical region, we have talked about this, where there is a locus which we define as a gene, okay, which is transcribed and translated. Fine. All I'm trying to tell you is the homologous chromosome will have exactly the same information, and it will have the same gene in the same location, which is pretty much over here. And these two copies are called alleles.
Okay, sir, got it. Thank you.
All right.
So in the next slide, when you said unit factors, was that meaning alleles or genes?
So here's where I'm not trying to cause confusion. The moment you talk in pairs, right? The pair is there are basically the same. It's the same information. So it's the same gene. When you say there is a gene for hemophilia, if there was a single gene for hemophilia, which there is not, you are talking about a single pair of alleles. So it's nomenclature.
Please repeat, your voice broke in between.
Okay, so what I'm trying to say is, let us say that there is a gene for which causes the disease hemophilia. Mutation in the gene causes the disease hemophilia. There are many genes which do that, but for the moment, let's just call it a single gene. And let's say that the gene is called H, okay? And this gene H will have two alleles in your body because you are a diploid organism. So there will be H and H, fine? And one H will be on one chromosome, the other H will be on the other chromosome. And this might be chromosome 21. I don't know exactly where the hemophilia gene is. Now, this is what we call as a wild type gene. If the gene gets mutated, then you will have, let's say only one copy gets mutated, then you will have two H's, but one H will have slightly different bases than the other H. So we call this we say one copy is mutant. If both copies of the gene are mutant, then we will say both copies of the gene are mutant. But their location doesn't change. And the fact that there are two copies of the same gene doesn't change. Clear?
Sir?
Yeah.
Can you please repeat what the inference we drew from the computer analogy of the genes, the hard drive one?
Right. So the analogy is only that I want you to imagine that. So what I'm trying to tell you is that all the information which I am using right now to speak to you, for example, is because of information which is coded in the genome of my body. Okay. My muscles are moving, my vocal cords are moving, I'm breathing. All this is controlled finally by the sequence of nucleotides in our genomes. So this sequence of nucleotides in our genomes is on a entity called as DNA. Fine. It's a double helix. It is just ATGC repeated all over again. you can actually type it out as English text. And if you typed it out as English text, because the information is the same, it's just chemical in your body and typing, but the content is the same, is basically packaged in distinct chromosomes. So this entire long, let us say 400 mbp strand is chromosome 1. And it's at its chemical nature, it is a double helix. So each strand has this information and the second strand is complementary to it. And this is one chromosome, this one long strand of DNA. You have a redundant second copy in your genome because you are a diploid organism. So there are 2 copies on your genome of the chromosome. Because there are 2 copies, at any stretch, whatever, if there is a gene coded by the stretch, the other chromosome will have exactly the same gene coded in that stretch. So the analogy is that these text files are nothing but information files, and chromosomes are nothing but basically packets of information, which if you have 21 chromosomes, instead of having one large file with all the information, you have 21 files in your body. That's all the analogy is.
Got it, sir. Thank you.
So what does this chromosome coloration indicate?
The red coloration?
No, in the top image, where it's written homologous pair of chromosomes and all.
Right. When the early cytologists, first saw chromosomes, they were not easy to see. It was only in the 1910s, 20s or 30s that people started seeing chromosomes. Making chromosomal preps was very, very difficult. And one of the examples is the fruit fly Drosophila melanogaster, which is the organism I do research on. And one of the things they saw in certain unusually large chromosomes was that if you stained it with a variety of stains, what would end up happening is you would see banding patterns on these chromosomes. Okay. So you stain one, you see one banding pattern, you see another stain, you see another banding pattern, and so on and so forth. So this is just a schematic representation of the fact that there were banding patterns. This band is basically area of the chromosome, which is nothing but a single double-stranded DNA, which contains, let's say, 10 genes, because chromosome is large. There are, let's say, 3,000 genes on this chromosome. So even this small stretch over here, which is showing a banding pattern or a staining, will contain 20, 30, 40 genes. Okay. So we know that there are banding patterns when we stain chromosomes. The reason for these banding patterns is not particularly important right now. Any other question?
Sir, is independent assortment only defined for genes or different chromosomes?
Well, the moment if you are working with flower position and pod shape simultaneously, right, you have a problem because they will not assort independently. So one of the major concepts in genetics is the concept of linkage. Okay, what linkage basically means, I'm simplifying it, is that any gene on a single chromosome is physically associated with all genes on that chromosome. And with exceptions, which you'll study about in genetics like crossing over, these genes travel as a group. So they are linked together. So the moment they are linked together, they will not show independent assortment.
Okay, sir.
So Mendel's rules will only work if you take one phenotype or one seed shape from one chromosome and pod color on another chromosome. So his famous dihybrid crosses will always be from 2 different chromosomes. He didn't know that, but that's how we are interpreting them today. Okay, so fine. So I assume you understand now from a modern perspective.
Sir, I have a very trivial doubt. During reproduction, one set of chromosomes comes from father cell and one set comes from mother cell. So,
Right.
sir, does that mean that information gets doubled at each generation, or does that mean that half of the information is lost during replication? I mean, during cell cycles or something.
So you have to remember that each chromosome, if you take one of these homologous chromosomes, if it is one of the 21 sets which you need to make a human being, one is enough. Fine. That is the reality. There is no law which says that you need both homologous chromosomes to make an organism. They are diploid organisms, they are haploid organisms. We are a diploid organism. So we have a double copy of chromosomes, one from mother, one from father, with pretty much the same information. Okay. If one gene is different from the other, then we say that the two alleles are not the same, they are different. So for whatever reason, the way evolution has decided our fitness, it seems that it is better for us to have two copies of the same chromosome, one from the father and one from the mother. But that's not true in animal life, in or plant life. There are plants which have tetraploid. There are plants which have 16 copies of chromosomes. There are animals, microbes out there which survive on a single copy of a chromosome. Okay. So what I'm trying to tell you is we have two sets of information. In theory, we can do with one. Is that clear?
Can we say that a chromosome has like some number of genes and each gene has some different characters?
In fact, that is the whole point, right? If you look at this graph over here, the humans have approximately, though it is still controversial, the number is reasonably accurate now. Humans have, let's say, approximately 25,000 genes. Okay. Now, each gene has a different sequence and it has different information. And that information is transferred through transcription to RNA. And RNA may be a functional entity, or mRNA, then mRNA makes a protein and the protein is a functional entity, right? So effectively, every gene is pretty much different. There are exceptions. There are duplicated genes which do almost similar work. But in general, let us say out of 25,000 genes, 23,000 are doing completely different functions. Did that answer your question?
Yes, sir. Thank you.
Sir,
Yeah.
I had a small doubt in the chromosome diagram.
Yeah.
Sir, in this slide, the chromosome contain two chromatids, but in left side of diagram, they said a single chromatid as a chromosome.
See, this is a chromosome. Okay. This happens only during DNA replication just before the cell is going to divide.
In for saying that they are homologous chromosome, there should be two chromatids, na? At least. So how they consider single chromatid as chromosome?
Who said so? This is a chromosome. If it duplicates, while it duplicates, it is still attached through the centromere, and we call each as a chromatid.
Okay, sir.
I'm not sure what the confusion is. If you look at the human karyotype over here, right? This is a karyotype. It's an actual picture of the way human chromosomes look. You can see that there are two homologous chromosomes over here. I think there is something with the schematic which is confusing you. Maybe we'll get back to that. Are there any other questions?
In that graph between chromosome and number of genes,
yeah,
the number of genes is exceeding the number of base pairs. So does that mean that some of the genes are overlapping in their codes?
No. It's not exceeding. This you have to. This kind of graph is something you don't see at all times. There are two axes, right?
Okay, I got
it. Where one axis shows
Pairs
one.
are different.
Okay, yeah. The point I want to focus on before I end this class is this idea of dominance. Now, all of you know that genes, there is a location on a DNA which is called as a D gene. The alleles are T or T. This DNA makes an mRNA. This mRNA makes a protein. And the protein is now made from either a T allele or it is made from a small t allele. Now, can you think of a reason why a TT combination does not give you any dwarf plants and why it gives you only tall plants? Can you repeat?
Protein which is formed from small t, it may be ineffective or maybe deformed in some way, but the protein from capital T is much more functional.
Okay, so let's take your example and I will say T makes an active protein. This is the simplest example, okay? And T is a gene which is, I'm going to use the term null, which is a genetic term, which basically means there is zero activity, fine? And I assign the capital T as 100% activity, fine? So now what is happening is the capital T, which is active, makes I'm again, don't take me seriously. I'm just saying it makes Horlicks, fine. And this is just to give an analogy that it gives some growth factor, which causes this plant to become taller than average, fine. So one copy of this Horlicks is enough, you get a tall plant. If you have a single copy, instead of having two copies, you still get the same height of the tall plant. You don't get an intermediate size. However, the moment you get two alleles which are not functional, you will not get a plant which is higher than a certain level. So if, let us say, call this 6 feet, this is 4 feet. So you either get 4 feet or you get 6 feet. Even if you have a single functional allele, you get 6 feet. The genetic term for this is haplosufficient, which means that to reach the maximum height possible, a single active T allele is fine. Okay. So the simplest explanation, molecular explanation for the Mendelian cross of Gregor Mendel, which has been reproduced many times, is that the small t is a dysfunctional allele. It does not give the growth factor. The big T is a functional allele. It gives a growth factor. However, a single copy is enough to reach the tallest plant. And when this growth factor is completely missing, you get a dwarf plant. Does everybody understand this explanation? Because I'm
Sir,
not...
I have a doubt.
Yeah.
If we say that T is like producing like 100% of say Horlicks, but like sir, double T should produce like 200%, right?
Exactly.
So like shouldn't that be like more than T, comma T?
So that is not happening over here. Okay.
But why does that not happen?
So one of the realities of genetics that, so for example, if you are talking of growth factor, right? If the growth factor needs to bind to a receptor, I'm using modern language, and that receptor has to signal to make, let's say, more cells or give more height, you have reached a threshold beyond which you are not going to get any taller. And it turns out that a single copy of T of that allele of that gene is more than enough to make you reach that height. Beyond that, putting excess is not going to make any difference. This is the way the genes are interacting in the pea plant.
So like this is due to feedback inhibition of sorts. That
it may be feedback inhibition, but the simplest explanation is beyond a point. Giving more Horlicks doesn't help.
Okay.
And not only that, you are not seeing any intermediate stages.
Yes.
So if you could have seen a 5 feet plant over here.
Yes, understood.
It would be additive the way you are suggesting it is, but we don't. So this is the way Mendelian discrete genetics is.
So a tall plant will get better sunlight, so it wants to grow tall. So capital T is dominating.
No, so you see, you are coming up with an explanation, which in this particular case has been already discarded. It turns out that this is purely a genetic phenomenon. This is, of course, sunlight is important. Of course, nutrition is important, but that is not, all this is done under equivalent conditions of sunlight and nutrition.
Okay.
Sir, how did Mendel make sure that in the first case he has two capital T's?
By having pure lines. So that is something you have to remember very clearly in your mind. Every experiment of Mendel was started with pure lines, which for generations bred true. So if you cross a red cow to a black cow, for example, to see what happens, for the 10 generations before, the red cow versus red cow mating only gave you red cows. In this case, tall plant versus tall plant mating only gave him tall plants. And he covered these plants, let's say, with plastic bags, and he kept them in a separate corner of his field so that there was no chance of any pollen coming from a short plant and contaminating his culture. In genetics, you have to define the potential alleles in your crosses before you start your crosses. Otherwise, things become very confusing.
Sir?
Yeah.
So can we say that the small t allele is not transcribed at all?
Is not transparent.
No, transcribed, sir. Transcription doesn't happen.
Which is what I'm saying. It's a null. It can be either not transcribed, or the mRNA may get degraded, or the protein may be made, but it's not folded properly.
Okay, sir.
Okay. So in genetics, we say that every allele has, let's just put a graph. This is where a single allele functions, fine, which is at 100. And at 0 is where what we call as a null. And anything in between, we use the term hypomorph. This is just terminologies. This is an important concept. What I have tried to do today is to define Mendelian genetics in modern terms. For those of you who already know all of this, refresh your mind in terms of genes, alleles, basic monohybrid Mendelian crosses. For those of you who are dealing with these concepts afresh, you have to sit down and think. They are not as simple as they seem. Mendel's ideas of segregation is basically alleles of two homologous chromosomes on two homologous chromosomes separating. Mendel's ideas of independent assortment have to do with the fact that genes are on different chromosomes and they don't influence each other. They are independent. Okay. And the third one, which is of dominance, is basically a concept of the fact that when there are two alleles and one allele is mutant, in this case, we are taking the simplest example of a completely loss of function null allele, then you basically do not see it in the first generation and it comes back in the second generation simply because two alleles and the laws of mathematics dictate that you will get the two null alleles back together in a ratio of 1:3.